Think we're going here. Yep, looks like it. All right, so plan today. Finish talking about chapter 8, get a start on chapter 9. Your final Master and His Emissary quiz is today as well. So those are the three big things for today. Anyone have any questions or comments about things before we get started? All right. So chapter 8, this is about the motor system. This is about the parts of our brain, the parts of the central nervous system that allow us to move. We've talked about, it's probably would be helpful to kind of review what we've talked about. We talked about the basal ganglia. We've talked about the cerebellum. And we've talked about primary motor cortex. We've talked about 3 brain regions so far. We'll talk about 2 more today, SMA, the supplementary motor area, and premotor cortex. These are both in the frontal lobe. In fact, I'll show you those. I've shown you this picture before, but just kind of refresh yourselves where everything is. SMA, supplementary motor area, PM, premotor cortex, MC stands for motor cortex, primary motor cortex. These are all located in the frontal lobe. The central sulcus, it appears in this brain, is about right here. The motor cortex is directly anterior to that. And close by are these other motor areas, which we're going to be talking about now. SMA, which is located more medially. So if you're measuring activity from the SMA, the best way to do it is just putting an electrode right on top of the brain, either towards the front or center of the brain, because the SMA is medial. It's towards the center part. It's close to the longitudinal fissure in the left or right hemisphere, so it's very close to the center parts of its brain. Whereas premotor cortex is located more laterally, more towards the sides of the brain. You can see that here, SMA towards the top center part of this brain, whereas premotor cortex is more lateral, more towards the side. Motor cortex, by the way, kind of runs the whole length of this. It's kind of a long segment. It starts up medially, but it ends up laterally. So motor cortex is kind of a long segment, where these other parts, SMA is more medial and premotor cortex is more lateral. And just so we don't forget, we've also talked about the basal ganglia, which is located kind of in the center part of the brain, sitting on top of the brainstem, on top of the subcortical areas. The cerebellum, which is right here. Brainstem, we actually haven't talked about too much. I've just mentioned that that's kind of an output towards the motor system. The brainstem works with the spinal cord, which is this thing right here, to eventually send messages to our muscles, which will cause our muscles to move. So those are regions of the brain we've talked about and are going to talk about. Once again, the ones that we're going to be talking about right now to finish off this chapter. The SMA, which is generally thought to be involved with voluntary actions, and I'll show you some neural evidence that supports this idea related to this thing called the readiness potential. And we'll also be talking about the premotor cortex. This is the one that's lateral, that's located in the sides of the frontal lobe, mostly thought to be involved with acting on something which is out in the world somewhere. So we've talked about another part of the brain, the dorsal pathway. The where pathway is important when we talked about this a long time ago. The dorsal pathway is a part of the brain that when you are trying to interact with some type of an object out in the world, the dorsal pathway, the parietal lobe, is involved with locating where that object is and the types of things that you can do with it. A related brain structure, which in theory is interacting a lot with the dorsal pathway, is the premotor cortex. It has very similar functions to the dorsal pathway, that is the parietal lobe. It's basically when you're interacting with an object, it helps you to have like a spatial idea of where that object is relative to you, and it gives you general information about the type of interactions that you can perform with a given object depending on where it is in space. I kind of think of these as SMA is like self-motivated actions when you decide to do something, where premotor cortex is more about how the stuff out in the environment is influencing your action. So this is more internal, this is more external. So there's a lot going on in this figure, but really all that, and this is taken from a Nature Neuro. This is taken from an actual article on something known as the they call it the readiness potential. But if you see down here in the slides, they call it, this is German, so I don't necessarily want to try and pronounce this, Bereitschafts potential. I think that's probably about right. Bereitschaft potential, which basically means the same thing as readiness potential. So on the test, really all I care about you knowing is the term readiness potential. It is measured with EEG. It is an event-related potential where you stick an electrode basically right in the center part of a person's head, right on top of the center medial parts of the brain, either kind of center or towards the frontal lobe. Presumably, we're measuring activity from the supplementary motor area, the SMA, when we're measuring the readiness potential. The way this paradigm works, the readiness potential paradigm works, is it's so simple. Basically, you are a participant. They're measuring your electrical activity with the electrodes placed medially, centrally on your head. And basically, they say something like, when you feel like moving, move. And that's where you get the voluntary aspect of this. You basically can sit there, not move at all, but then all of a sudden you're going to get the feeling to move. And I want to make sure people can see this. It's not showing me. Okay, well, yeah, there we go. And so you'll be sitting there and you won't be moving, and then all of a sudden you'll get the urge to move, and they'll be measuring your brain activity, and they'll be like, move your hands when it's time for you, when you decide you want to move. But it's completely voluntary. It's your decision. It's an internally decided thing. When you feel like moving your hands, move your hands. And what they're doing is they're looking at the neural correlates of this. How does the SMA, this region that they appear to be measuring when they place the electrode in the front to center medial parts of the brain, how is this responding when you're making this decision to make a voluntary movement compared to when you are not making a voluntary movement? So the first thing is that, so time point 0, as you can see right here, time point 0, this is when you decide to make the movement. Time point 0. So the event-related potential is measured in response to your voluntary movement. What's really interesting about the readiness potential, though, is that usually when we're measuring event-related potentials, the electrical activity that we're interested in comes after a stimulus is presented, after some event. The readiness potential is one of the exceptions. It's one of the few event-related potentials where the really important stuff actually happens before the event. Once again, time point 0, that's when you decide to move. That's when you decide to move your hands. And then what you notice is that when that happens, there's actually a drop in activity. And the actual drop, that's a positivity. This building phase right here, it's negative, negative, negative. And you can see that because negative is up here. It's getting more and more negative up until the point in which you decide to make the movement, in which case it drops back down to the baseline levels. And so this stuff here, this is the stuff which is of most theoretical interest to researchers. What is happening up until the point that the movement is made? And what's interesting is that before you actually make the voluntary movement, something about your brain knows that it's coming. As far as like maybe 1 second before you actually make the movement, there's this building process right here that keeps getting more and more negative up until the point in which you actually make the movement, at which things drop again. So this is kind of interesting because it's like for at least 1 second before you actually make the movement, it's like your brain knows, okay, I'm going to be making the movement soon. I'm going to be making the movement soon. And it's getting ready. That's why they call it the readiness potential. Up until the point that the movement is actually made, at which the activity becomes more positive again. So this is the readiness potential. It's a increasing negativity up until the point of the movement, which then drops back down to baseline levels. And the really interesting thing is the building negativity that occurs before the action is performed. Questions or comments about readiness potential in the SMA, supplementary motor area, which is thought to be underlie this effect?
Is this what McGilchrist was talking about?
Yes, it is. Yes, it is. Exactly. Now, he was talking about a more specific study, the one with, I believe, if I'm recalling correctly, he was talking about the one where there was a clock and people were looking at the hand of a clock. So I'm not going to get too much into that one. Yes, he was talking about the readiness potential, but he was talking about a very specific study there's like hundreds of studies that have used the readiness potential. He was talking about a particularly famous study which used the readiness potential and was used to make these arguments about free will. The basic idea, without going into the details of what McGilchrist was saying, he was basically saying that there are parts of our brain that know that we are going to make a movement before we are consciously aware of that we are going to make a movement. So it suggests that some aspects of our voluntary movements are actually unconscious, that we don't have complete control over them. Because according to that one study in The Master and His Emissary, can you mute that microphone, please? Whoever that is, I guess I can probably do it myself. Yeah, please mute your microphones there, people. Yeah, but McGilchrist suggested it was an unconscious, that our brain knows that we're going to move before we are actually aware that we are going to move. Any other questions or comments? All right, so here's the premotor cortex now. Yeah. I still hear a microphone, so if you people out there could mute your microphones, I'd appreciate it. Okay. Just gonna have to go through and see who that is. Just don't know. Okay. Yeah, please mute the microphone. I can't tell who that is, but whoever just coughed, we could hear you. So please mute that microphone. Okay, that's a little better. So the premotor cortex, that's what we're talking about here. We mentioned this is externally guided actions. So this appears to be a part of the brain which represents the potential points of space that you might be interacting with in the near future. That's what this study basically says, is that, well, here they're measuring 2 cells. Each cell is known to have like a preferred point in space. So earlier we've talked about preferred direction. These cells have like a preferred point in space, or a specific receptive field that they respond to. So this cell, which is colored, which is red here, this responds to like the upper right-hand point of space. And this blue cell, so the red cell is all of this activity over here. The blue cell responds to this lower left point of space right here. And you can see this is the activity, of course, across the trial of this one cell. So we have these 2 cells, and they respond to different points in space. And in this study, they're looking at how the activity is influenced by the potential points in space where a target might occur. So at the beginning of this experiment, or the beginning of a given trial, in this particular trial, the participant will see 2 spatial cues. And what's that's telling the participant is there's 2 possible places that the target could appear. By the way, the target, which they call the go signal, is this thing right here, which is occurring in the upper right side of space. But at the beginning of the trial, you're given 2 possibilities of where things could occur. And so one of the questions is, well, this is like a preparatory interval right here, or a delay interval, because they've given you a cue, and then you're waiting for your, looks like 500 milliseconds before you are given this color cue right here. And what this color cue is telling you is this is telling you exactly which part of space this thing is going to be in. And if it's a red, then you know it's going to be in the upper right-hand point of space. If this thing was blue, then it would be in the lower left-hand point of space. And so a couple things to notice is that during the preparatory interval, both potential points of space are kind of simultaneously represented. It's like these 2 potential actions. One action is you're supposed to respond to the thing in the upper right-hand corner. The other potential action is responding to the target that might be in the lower left-hand corner. The blue responds to the lower left, the red responds to the upper right. And during the delay period, both of these neurons are showing small amounts of activity, not tons and tons of activity, but this is definitely above baseline activity. There's some activity here, which is kind of interesting because this suggests that when both of these things could happen in the near future, your brain is kind of preparing to respond to either one. This is how your brain appears to deal with ambiguity. Am I going to do upper right hand or upper left hand? It kind of represents both to a small degree during the preparatory interval. But then as soon as you're given the color cue, which tells you exactly where the target's going to occur, you notice how the response on this neuron responds to the lower left-hand part of space, that is the blue cue. The activity drops drastically there while the activity increases here. So once again, this is just another region of the brain that appears to represent in particular spatial locations of things so that you can interact with those things once they actually appear. This particular study is interesting because it shows these anticipatory effects during the delay interval, both potential actions that you could perform, that is interacting with the thing in the upper right or interacting with the thing with the lower left, both are kind of represented before you know for sure where the target's going to appear, in which case one neuron wins, so to speak. The upper right-hand neuron wins and gets a lot of activation, where the upper left-hand neuron gets very little activation. Any questions or comments about the premotor cortex versus the SMA we just talked about? Cool. Let's go to chapter 9 then, and we'll do a little bit of stuff to get started on chapter 9, then we'll get your master and emissary quiz started. All right. Now talking about memory. Fun chapter. Memory is a very good topic for cognitive psychology and cognitive neuroscience. And here we are talking about kind of the standard model. I guess I'm showing you two different pictures. This is from a cognitive psychology textbook right here. This is, I think I probably got this from Google. This is showing a more detailed version of what's known as the same basic model right here. This is showing a slightly more detailed model, a famous one called the Atkinson and Shiffrin model. And basically what both of these pictures are doing is that they're showing, in theory, what the three components of memory are. And the three components of memory are thought to differ primarily in the amount of time information can be stored in each component of memory. There is a couple exceptions about how these, each component has unique properties other than just the duration that information could be stored. But let's first talk about the duration of time the information can be stored in each. Sensory memory, by definition, is like the first aspect of memory that is stimulated. When you're taking information with your eyes or ears or any sense, sensory memory kind of represents that raw sensory data which is coming in from the world, but it does so for a very short period of time. That's really the crucial theoretical property of sensory memory, is that it's representing information which is coming in via some sensory receptor, but it does so for basically a fraction of a second, or at least a very short amount of time. Compared to short-term memory, which is able to represent information usually along the lines of, maybe up to 30 seconds or a minute is the typical kind of cognitive psychology explanation, maybe up to 2 minutes. And then long-term memory, which can represent information for a really, really long time, maybe up to your entire life, like years and years and years. It might be information which basically will last you forever. For instance, my example of long-term memory, I still remember the telephone number 253-3058, which is the number of my house when we moved out of that house when I was 4 years old. So that was 34 years ago, yet I still remember this, which shows that like my long-term memory works okay. My short-term memory, long-term memory definitely seems to be working okay though. So these are the differences in the duration. Sensory memory is very quick in the duration of information stored. It decays very quickly. Short-term memory is a little bit longer. Long-term memory lasts the longest. Another thing though, which is really important about short-term memory, is that it's got this kind of loop right here where information can leave short-term memory and go back into short-term memory, and this is typically known as rehearsal. So this is the idea that, we don't have to do this too much anymore because now you can type in numbers into phones really easily. But back in the day, before people carried around mobile phones with them all the time, someone would give you their telephone number. And if you didn't have any way to write it down, what you would have to do is you'd have to say the number in your head over and over and over again until you reached a piece of paper, or until that information stayed in your long-term memory. So rehearsal is an example, would be like someone gives you your phone number and you kind of say it to yourself, 253-3058, 253-3058. You say it enough times inside your head, in your mind's ear, and until you find a piece of paper where you can write down the telephone number. Or if you say it enough times, maybe that information will actually be, you can see this arrow right here, will actually transfer to your long-term memory, where that will stay there permanently, or at least for for a long amount of time. Another thing about short-term memory is that it can interact with long-term memory in these kind of bidirectional arrows, and we will be talking about this. Information can go from short-term memory to long-term memory. The first time I tell you a telephone number, you're using your short-term memory to kind of rehearse that information. But if you say it enough times, or if you've dialed that number enough times, eventually it goes to long-term memory. But there's this other arrow which goes back from long-term memory into short-term memory. This has to do with the fact that short-term memory is often assumed to be the point in your mind, the point in your memory system where consciousness occurs. So when I take that number, 253-3058, I haven't thought about that number since I taught my cognitive psychology course Monday, because we were talking about memory there. And so that number, 253-3058, it's in my short-term memory right now because I am conscious of it. But I haven't been conscious of it since Monday. That was the last time I was conscious of it. So what I just did is I retrieved information from my long-term memory and brought it into my short-term memory so that I now have that telephone number, 253-3058. It is being represented in my short-term memory right now. And when I'm done talking about this, it'll go away. I'll no longer be consciously thinking of 253-3058. And then the only activation for that will then be in long-term memory. So short-term memory, long-term memory have some interesting components. Short-term memory has this rehearsal thing. And then the way these two components interact, information from short-term memory can be transferred to long-term memory, will be stored permanently, or at least for a long time. But information can also be retrieved from long-term memory and brought into short-term memory, which is where conscious awareness of your long-term memories would occur. So whenever you're thinking about a memory from your childhood, you're bringing that memory and you're conscious of it, you're bringing that information into your short-term memory, and you're doing so by retrieving it from long-term memory. Any questions or comments about these three basic memory systems? All right. Okay, so we're going to go a little bit more about sensory memory. Actually, really what we're going to talk about the most is echoic memory, which is a type of sensory memory. So typically, sensory memory is kind of divided up into visual sensory memory, which is known as iconic memory, versus auditory sensory memory, which is typically referred to as echoic memory. The thing that these both have in common is that the decay is quick. However, in the visual system, decay is thought to be incredibly quick. So we talk about this idea that there's a sensory memory, and you look right here, there's a single sensory memory box, and, in this model, there's a single sensory memory box. It's a little bit more complicated than what these basic models, like the Atkinson-Shiffrin model is showing, because in reality, there's probably kind of two separate sensory memories. There's one for visual, and there's one for auditory. The visual version of this, the visual sensory memory, is called iconic, and it's thought to have very quick decay. So we're talking about decay along the lines of like 500 milliseconds. So you see something with your eyes, it's represented very, very quickly with sensory memory, like 300 to 500 milliseconds, and then the sensory memory image goes away. One way of kind of thinking about this, it's kind of like a metaphor, an analogy. When you wave your hand in front of your hand very quickly, you see little tracers a little bit. I don't think I'm just hallucinating or anything. I think those things are really there. Most people appear to experience them. Potentially that those little tracers you see when you move your hand in front of your face is iconic memory. It is the persistence, the fact that there's kind of this very quick decay time that's like along the lines of milliseconds, maybe 200 to 500 milliseconds. And as your hand moves, the information is kind of decaying, and that's what you're seeing. You're seeing the decaying information, the fact that it takes about 200 to 500 milliseconds for that visual information to decay. That's iconic memory. Very, very short persistence, very quick decay times. Compared to echoic memory, which the evidence is pretty clear here that echoic memory, the decay time is longer, along the lines of seconds. And I'll show you some evidence. I think I'll show you that evidence next time. But the evidence is that the decay time so there's longer persistence, longer decay time in auditory sensory memory compared to visual sensory memory, where the decay time is very quick. And auditory memory, the decay time is a little bit longer. I kind of like the analogy of echoic memory to kind of like being in a cave, how you say echo, and then it echoes what it says, the word, you hear the cave, repeat the word echo back to you. That's kind of the way echoic memory is a decent metaphor or analogy for echoic memory. It also kind of captures the idea that whereas visual sensory memory is incredibly quick, there's almost no echo at all, or it's a very, very small echo. for echoic memory, auditory sensory memory, echoic memory, the decay time is relatively longer, kind of like the echo in a cave. My absolute favorite illustration of echoic memory comes from the Cowan task. And so, I wonder if I wrote the instructions here. I didn't. You should write this down. Oh, no, it says it right here. Repeat as many of the digits at the end of the sequence as possible. So what I'm going to do is I'm going to read off a long sequence of numbers to you. And your job, the sequence is going to be so long that you wouldn't possibly be able to remember the entire sequence of numbers. It's just going to be way too long. You couldn't possibly remember the entire sequence of numbers. So instead, what I want you to do is what's repeated here. Remember as many of the digits at the end of the sequence as possible. Technically, the participants repeat them, but I don't want a bunch of people repeating stuff randomly when we're doing this. So just kind of write them down. Once I'm done saying the sequence, write down as many of the digits at the end of the sequence as possible. And the goal is to remember as many of the sequence, many of the digits, many of the numbers as the end of the sequence as possible. So let me write down some of the sequence here. Good, I'm ready to go. So once again, I'm going to say a long sequence of numbers. And your job is to remember as many of the digits as many of the numbers at the end of the sequence as possible. Once I'm done reciting the sequence, write down as many of the numbers you can at the end of the sequence in the correct order, by the way. Okay, so here goes. 4, 2, 9, 6, 1, 8, 7, 8, 3, 2, 6, 9, 5, 7, 8, 6, 4, 2, 1, 9, 6, 4, 7, 8, 2, 1, 6, 4, 3, 8, 9, 2, 6, 4, 7, 5, 8, 1, 3. Write down as many of the numbers at the end of the sequence as possible. Okay, so I always am reminded of The Price is Right, the game One Away and The Price is Right, where they say, ladies and gentlemen, I think it's just only women judges now. Ladies, do I have at least one number right? I imagine everyone has at least one number right. Let's start with, what were the last three numbers? What were the last three numbers? Does anyone know? Ladies, do we have at least three numbers right?
I said 813.
That is correct. 813. So we have at least three. That is correct. Do we have anyone who got the last four numbers right? Is three all we got? Anyone think they have those?
4813.
It is not 4813. Don't feel bad, by the way. I always feel like I need to tell everyone this. My wife and I did this when we were traveling from Las Vegas to California once, and if you've ever done that drive, it's like a 4-hour drive, and it's very boring because it's desert, like for 3/4 of the way there. So we were very bored, and we were actually doing this task, the Cowan task, back and forth to each other, and I never got more than three. Not one time did I get more than three. And I'm very competitive. My wife got 4, sometimes 5, and so that may be a little bit mad. But I guess what I'm saying is 3 is pretty typical, and if you got 3, you did as good as me. Anyone got that fourth number, though? The last 3 numbers were 813. Anyone think they know what that fourth number was before the 8?
Was it a 5?
It was a 5. 5813 were the last 4 numbers. Now, does anyone think they know the last 5 numbers? Anyone think they got the last 5? So we know the last 4 numbers were 5813. Does anyone know what number came before the 5?
7.
It was 7. This is good. One more and you win the new car. Actually, in One Away, there's only 5 numbers. So you just won the new car from The Price is Right. Good job. But let's still continue to see if anyone can get, did anyone think they got the 6th number? So we know the 5 numbers where the last 5 numbers were 75813. Does anyone know the number that came before the 7?
Was it 6?
It was not. You're close, but it was not 6. It was 4. Did anyone get that and just not brave enough to say it? The last 6 numbers were, I'll just tell you the last 9 numbers. 9, 2, 6, 4, 7, 5, 8, 1, 3. So the idea here is, now it's hard to, usually in class I'd have people raise their hand, but you guys probably got a feel. I think no one got that 4, so no one got 6 right, unless someone did and they just weren't brave enough to say anything. So the highest answer was 5, and there weren't a whole lot of people in the class that got 5 numbers right. Most people on average, by the way, get 4. So I was slightly below average when I did this with my wife on that road trip, with getting about 3. The average, though, is 4 with a margin of error of about 1. So 4 plus or minus 1 is pretty typical. So basically, typical performance on this is between 3 to 5 digits to be remembered on each trial. And this is thought to be a measure of echoic memory, that is auditory sensory memory. And the reason is, it says right here, the sequence length is unpredictable. You didn't know when the sequence was going to end. Therefore, it eliminated a strategy that you oftentimes will use with your short-term memory, and that is rehearsal. You were not able to rehearse all of the numbers as I said them, because as soon as you start rehearsing numbers, I'm going to be listing off a whole bunch of new numbers. You didn't know which numbers to rehearse. So the typical strategy for people is they kind of just wait passively until the sequence is done. And then when they're done, there's kind of some numbers that are echoing in their head, and those are the numbers that they are able to report. The longer the echo is in your head, the more numbers that you're going to be able to report. Me personally, my echo is pretty short. I was only able to get about 3 numbers, though I have had a couple students who have been able to get 7 digits, but that's freakishly good echoic memory capacity. Typically, once again, it's 4 plus or minus 1, because the length is unpredictable, which means that you cannot use rehearsal, or it makes it much more difficult to use rehearsal to perform this task. So that's what's cool about this Cowan task, is that it's thought to be somewhat of a pure measure of echoic memory. It kind of eliminates your ability to use your short-term memory because you don't know when the sequence is going to end. So it's thought to be just that echoic component of memory. Questions or comments about the Cowan task? Oh yeah, so we will be talking about this. Yeah, so here's more evidence that echoic memory is around. I said the echoic memory was longer than iconic memory. And here is evidence that echoic memory has at least a duration of about 3 seconds, according to the study. So this is using an event-related potential. Actually, I think in this study they were using MEG, magnetoencephalography. We've usually talked about this with respect to EEG using electroencephalography, but they're really the same. There's really not a whole lot of difference between them. But we're looking at the mismatch negativity, or synonymously called the mismatch field response. And to remind you how this is an event-related potential. And the way this works is that there's a standard and a deviant stimulus. And so the standard stimulus might be like the syllable ba, and the deviant might be the syllable da. And you say the standard a whole bunch of times, and then 1 out of like 10 times you'll say the deviant. And the mismatch negativity response is the idea that you generally will see a bigger negativity for the deviant. And you see that right here. And then, by the way, the mismatch negativity response usually has a latency of about 100 to 150 milliseconds. So right here, you can see that the blue and red lines are separated. That is the mismatch negativity response right there. The blue line has more of a negativity relative to a red line. The blue line dips down below the red line right here. That is the mismatch negativity. The blue line dips below the red line here. That is the mismatch negativity. The blue line dips below the red line here. That's the evidence of the mismatch negativity. But if you look here, you might notice there's no difference between the blue and the red line. There's no difference between the blue and the red line. There's no difference between the blue and the red line. And so we want to understand, why is there no difference between the blue and the red lines for these things where there is here. So we need to talk about what all these different things are. And if you look right here, it says interstimulus interval. Interstimulus interval means the amount of time that occurs between each stimulus in the experiment. So I'll first start with a very, very fast interstimulus interval, and I'll do several of them since this will help illustrate what's going on in the experiment. Ba, 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 da, ba, 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 da. So that was relatively fast interstimulus interval where I'm saying one word, more than one word per second. I'm saying like 1.25 words per second, or maybe, the interstimulus interval was somewhere around 3/4 of a second for what I was just doing, I think. When its interstimulus interval is really long like this, there's a much more time that elapses between each stimulus. So that would be like ba. Wait 12 seconds. Ba. Wait 12 more seconds. Da. So on and so forth. So I'm not going to do a whole bunch of those because that would take a really, really long time to do that. But what you notice here is that you see this difference between the standard and the deviant when the interstimulus interval is, well, you tell me, do we see more of a separation between those blue and the red lines when the interstimulus interval is small or when the interstimulus interval is long?
Small.
Yeah, small is correct. You basically see a difference between those red and blue lines for these conditions right here. These are the ones where the interstimulus interval was small. There's basically no difference between the red and blue lines here when the interstimulus interval was big. So a couple things. There's a couple reasons to believe that this is echoic memory. And a couple properties of the mismatch negativity have led people to believe this is echoic memory. The main thing is that in the mismatch negativity studies, oftentimes people are performing some type of distractor task. Like usually the main one is they're watching a silent movie. So they have headphones on and they're listening to the bahs and dahs in their headphones, but they're watching a silent movie and they're actually told, don't pay attention to what's going on in your ears. And so if they're not paying attention to anything, then in theory, they're not using your short-term memory, because when you're doing like rehearsal and those types of things in short-term memory, that's thought to rely on attention. If you're not paying attention to the sounds, then you're not using your short-term memory to keep them activated in your short-term memory. You're paying attention to this silent movie and you're ignoring the sounds completely, which means that the sounds should only be processed by your echoic memory. They shouldn't be traveling to your short-term memory once again, because you're ignoring them. You're not paying attention. The only things in theory that can travel to your short-term memory is stuff that you're paying attention to. If you're paying attention to something else, if you're ignoring these sounds in your ears, then the information shouldn't get to short-term memory. If you make that assumption that these neural responses are measuring echoic memory and that short-term memory has been erased or is not included, then these findings do seem to suggest that the duration of echoic memory is about 3 seconds. The idea here is that what the mismatch negativity response is doing is it's taking a representation of the standard, which is maintained in your echoic memory, and then that information decays. Some amount of time that decays, but it's there for a little bit. And when a new sound comes in that's a deviant, your brain detects the mismatch. That's what the mismatch negativity gets that response. When the standard and deviant are different, in echoic memory, that's the part of your brain which is able to detect the mismatch. When there's a ba stored in your echoic memory, but then there's a deviant sound, da, it's deviant in the sense that it's different than the other sound. Your brain detects the mismatch, and that's what causes the negativity for the blue stimulus. That is what causes the negativity for the deviant stimulus. If the sound in your echoic memory decays, though, then when the deviant stimulus comes, there's no mismatch anymore because it's comparing itself to nothing. And so there's no mismatch because the original stimulus that had been represented in echoic memory has decayed. So there's no mismatch. In order for there to be a mismatch, you have to have both of those stimuli represented in your brain. But because of the decay of echoic memory, especially for these long interstimulus intervals, the ba sound comes and then it decays. And then the da sound comes later, but there's no mismatch because the ba sound has decayed away from echoic memory. In these short interstimulus intervals, though, you hear the ba, and before it can decay, the da sound comes, and you can hear and the brain detects the mismatch. So kind of the punchline of this study is it suggests that the duration of echoic memory is up to 3 seconds, which is definitely longer than what has been postulated for iconic memory. Visual sensory memory or iconic memory has been suggested to have decay times of milliseconds, like 500 milliseconds at most, maybe 300 milliseconds, while echoic memory, 3 seconds would be like 3,000 milliseconds. So definitely longer persistence, longer decay in echoic memory compared to iconic memory. Questions or comments about this study? Okay, I think there's just one more thing to talk about in this chapter, and then, then we'll call it a day. And that's the Miller task. So I've already shown you 2 pieces of evidence that related, to this idea of echoic memory. The Cowan task was a task which measures echoic memory, which suggests that echoic memory is usually able to remember about four things. The mismatch negativity study suggested that the decay time of echoic memory might be around 3 seconds. And so that's 2 pieces of information that I've given you about echoic memory. Now we're going to contrast echoic memory with short-term memory. So echoic memory is the part of our mind where the decay times are relatively quick. Short-term memory is the part of our mind where the decay times are relatively longer. And to understand this, a good comparison is to compare the Cowan task, which we've already talked about, more of a pure measure of echoic memory, to the Miller task, which is it's a task, it's actually pretty, also famously known as the digit span task, where in the Miller task, some of you might have heard in your intro psychology class, this is the one where the famous study where it says Miller's magic number 7 plus or minus 2. I imagine some of you guys have heard about that. Miller's magic number 7 plus or minus 2, which is oftentimes thought to be the capacity of short-term memory. The way the Miller task works, which is described here, is that you're given a short sequence of numbers, like 5 to 9. And your task is to remember as many of the sequence, your job is to remember basically the entire sequence. So in the Cowan task, you're asked to remember just as many numbers at the end as you possibly can. In the Miller task, you're asked to remember the entire sequence. But in the Miller task, the length of a given sequence is somewhat predictable. It's going to be somewhere between like 5 and 9 digits usually. It's not going to be like this 40-digit sequence like it was in the Cowan task. The Miller task is going to be much more manageable. Your ability to actually remember the whole thing is better. So let's do that. Let's do one. Okay, so here's a sequence that I've just made up, and I want you guys to try and remember all of the numbers that I'm about to give you. So maybe write them down once I'm done with the sequence, and then we'll play the one-away game from Price is Right. Okay, so here we go. 9, 3, 6, 2, 1, 4, 5, 8. Anyone think they have all 8 of the numbers?
Is it 9, 3, 6, 2, 1, 4, 5, 8?
Yes, very good. That's good. That was 8 numbers, and you got all 8. So what I want to point out here is that with this task, we very quickly got an example of a student that was able to remember 8 numbers. Remember, we did for the Cowan task, not a single student got 7 numbers right that I'm aware of. The highest was 5 or 6. I don't remember now. Maybe it was 5. Or 5 or 6 was the highest any student got in our class for the Cowan task. For the Miller task, we showed right away that a student was able to get 8 digits, no problem. And so what this shows pretty unambiguously, and the data definitely support this as well, is the Cowan task is harder than the Miller task. The reason is, at least in theory, is that the Cowan task is isolating echoic memory. The Cowan task eliminates your ability, it prevents rehearsal, and another thing that we'll talk about more next time called chunking. The Cowan task does not allow you to use your short-term memory. The Cowan task, echoic memory only. The Miller task, on the other hand, does allow you to use chunking and rehearsal, these capacities which are thought to reside in short-term memory. When I said that sequence right there, as soon as I say the first number, you can just start rehearsing each number as it comes along. And by the end of the sequence, you're kind of rehearsing all of those numbers in short-term memory. which explains why your performance in the Miller task is better. Any questions or comments about the difference between the Cowan and Miller tasks? So I do want to talk one more thing here. Actually, no, we'll do this next time. I'll let you get a start now. We'll talk about hemispheric asymmetry for short-term memory of verbal and spatial information starting next class. So now I'll stop sharing. Does anyone have any questions about the material before we start the quiz? Stop sharing now. Or I'll stop recording now.